Yeah, we can hear you. Great.
Okay, great. Thanks, Clive, for the invitation to join you all, and I'll echo everybody else in congratulating you for 20 years of Immunopaedia, and I really watching the impact of that over the last few years, particularly, has been just amazing to watch. I added the slide right at the very last minute because I was listening to the earlier talks and listening particularly thinking of impact to the talk from Tammy Myers right at the very beginning. And she mentioned that about 20 years ago, she was working at Barra, and the impact of her research and that of others in the room on preventing HIV in kids who were being born in that hospital at the time. And I kind of made me quite emotional because two of those kids are on the slide with me here. These are my sons, both of whom were born at Barra in approximately those years. And if you want to see the impact of immunology, then I guess for me, there's nothing more than seeing these two boys who are now smelly hormonal teenagers who were saved by those kind of interventions. So my additional emotional slide before I launch into science. For the rest of my talk, I will be a little bit more grown up, I hope, and talk about advances in the HIV vaccine field. And I changed my slides right at the last minute to make them more general because I realized what a general audience we were speaking with. So I'm going to take you through where I think we are in terms of HIV vaccines, what the big challenges are at the moment and what the big successes are at the moment, and very much in the context of recognizing that a lot of what I'm speaking about has been severely compromised by some of the recent funding cuts. But I hope I can persuade you that there's a lot of good science that still needs to be done in this field. Of course, the major problem in HIV vaccines is the level of variability. And this is a slide that compares variability in SARS-CoV-2 with variability in HIV. And I think probably it's the most humbling reminder of what the challenge is with HIV. With SARS-CoV-2, we recognize how diversity within the virus made it difficult for the vaccines to keep up with emerging variants of concern. On the left-hand side is a highlighter plot, and on the right-hand side, this little cloud of both of which summarize diversity within SARS-CoV-2 pre-Omicron. And down at the bottom is global diversity in HIV. And you can see it's orders of magnitude difference. And I suspect most people in the room know this, but I think it's an amazingly visual way of highlighting why it's been so difficult to make a vaccine to HIV, despite the fact that we use many of the technologies that have been developed in the HIV field over the years to make the SARS-CoV-2 vaccine. I've spent those 20 years, more than 20 years that Clive referred to, basically studying 4 or 5 people who developed broadly neutralizing antibodies to HIV, and then trying to translate what we understood from those people into vaccines. A lot of the work I've done over the years has been nested within the Caprisa cohort that some of you may be familiar with in KZN. And many years ago, we recognized that understanding how people's immune responses to HIV infection matured could inform how to trigger the same kinds of responses by vaccination, which is ultimately our game. But even within people living with HIV, very, very few people make what we call broadly neutralizing antibodies, which are the types of antibodies that we will need in an HIV vaccine because they'll be able to deal with the level of variation that I showed you on the previous slide. And only about one in 5 people make those antibodies. And unfortunately, those people who naturally live with HIV and develop broadly neutralizing antibodies do no better than other people living with HIV, largely because it is the virus itself that drives those responses in people. But of course, the premise of everything that I'm going to speak about today, vaccination and now passive immunization, is based on the idea that if we had those antibodies present in people before the virus through vaccination, then that would yield a very different outcome. So we've spent, this is kind of 20 years in one slide, and we and others have spent a lot of time understanding these elite neutralizers and studying their B-cell responses, studying the viral evolution that happens in parallel. Kondwani spoke a lot about the trainees, and this one individual has been the basis of many, many degrees, some of which are shown in this slide, and many, many collaborations, both within and outside of the African continent. But we now have, from these kinds of studies in the Caprisa cohort and in others, an understanding of the roadmap that drive some antibodies from being kind of strain specific and not very potent to being much broader in the types of antibodies that we would want to see amplified by vaccination. And understanding that has been very much the basis of what is now referred to in the field as rational vaccine design strategies. This is a schematic from Bart Haynes and colleagues describing what rational vaccine design strategies are. Whenever I present this slide, I kind of think, well, because it does beg the question of whether everything that we've been doing up to now has not been rational in 20 years of research. But I think the difference here is that what we're trying to do is much more carefully, by very specific engineering of both precursors and of vaccines, try to copy the process that happens in people living with HIV, rather than using some of the empiric vaccine design principles and processes that have failed so far in vaccine design. I'm going to try to take you through in this talk some of the concepts that I think are changing the HIV vaccine field. And the reason I'm focusing on the concepts is because I think they all have utility for the design of vaccines and preventative antibodies for multiple different pathogens, particularly the very variable pathogens. So I'm going to speak a little bit about progress in germline targeting, some work we're doing immunizing people living with HIV, some data from pediatric trials, and then speak a little bit about slow delivery immunization, and try to highlight as I go how much of this work is being done on the African continent by African researchers. Germline targeting has been something that's been very much in the HIV vaccine field news over the last couple of years. And what this is this recognition and understanding from our studies of people living with HIV that the precursors of broadly neutralizing antibodies are structurally and genetically unique. In many cases, they have either very long CDOH3s or very short loops. In many cases, they use very rare germline genes. And the idea behind germline targeting is understanding that what makes those precursors special means that we can engineer vaccines to specifically recruit those precursors and to trigger them by vaccination. And the idea is to enrich what would be otherwise incredibly rare precursors because of those funky genetic features in our naive B cell repertoires. In general, these precursors are so rare that maybe the average person would have somewhere between 1 and 5 B cell precursors. So it really is looking for a needle in the haystack. And this work has really been led out of the US by investigators like Bill Sheaf, Leah Stamatatis, and then Roger Sanders in the Netherlands. But over the last couple of years, since its first paper in Science in 2023, triggering of these precursors has finally become a reality. And these kinds of highly engineered vaccines are now able to activate these precursors, and more importantly, to drive them towards some degree of somatic hypermutation, picking up the kinds of mutations that we know from the studies of people living with HIV are important for those individuals in order to develop breath. But I really wanted to highlight, and this is where there's a key gap in the field, and I think where the African HIV research community can really make useful contributions. There's a key gap, which is that we need to take these activated precursors substantially further along the maturation process. They need to pick up a whole lot more somatic hypermutation, and we need to drive much higher levels of these antibodies within plasma for us to get even close to having a protective HIV vaccine. I'm going to speak a little bit more later in the talk about some of the opportunities that we have here to accelerate this kind of research. And some of this is already being done by people like Eunice and Duarte and Daniel Moema and others in Kenya. So very much in place and set up, ready to go here in Africa. But measuring success in these kinds of trials is really difficult. And Daniel and Eunice and others would confirm this. They're not online. But measuring success in germline targeting trials requires next generation deep sequencing. It's very different to the traditional vaccine trials where we can measure neutralizing antibodies or binding antibodies, kind of low tech, high throughput assays. In order to know whether germline targeting vaccine strategies have worked, we have to sort out individual B cells and sequence their repertoires. We have to understand the degree of somatic hypermutation and the targeting of those antibodies and understand the germline genes that they came from. And I think the fact that this can only be done by deep sequencing probably speaks more than anything else to the fact that we still have a very long way to go in these vaccinees. If we can only pick up these responses by deep sequencing, then for them to reach the level of protective titers that we think we need, there's a very long way to go. And these kind of trials are underway at the moment still in Africa. The germline targeting idea is very much based on the fact that many people, unrelated people from diverse populations across the world, mount what's called a public antibody response, which means that the antibody response I mount to this epitope would be very, very similar to the antibody response that anybody else in the world might mount to this particular epitope of HIV. And that's shown on the right-hand side here, a structure of HIV at the top, and antibodies from 5 different donors overlapping in their binding to the envelope. And you can see that they're virtually superimposable, despite the fact that they come from genetically different populations, from people across the world, some African, some European. Everybody responds in the same way for this particular class of epitopes. The problem is that most broadly neutralizing antibodies to HIV don't have such a clear genetic signature. This is by far the clearest and most conserved approach to the envelope, to the HIV envelope. But there is preliminary data from the CHARD consortium that many of you will be familiar with, showing that you can potentially expand germline targeting to lots of different epitopes. As you know, the CHARD consortium is one of those that have recently had their funding significantly cut. But if anything, that is a place where we need to step in rather than step away, I believe. We have to work with folks who've developed these kinds of vaccines to get them into people and generate the data we need in a much more constrained funding environment than we were working in 6 months ago. I added this slide also while Kondwani was speaking, because Kondwani mentioned the importance of understanding African genetic diversity. And I completely agree with Kondwani that we have much too little understanding of this at the moment. Certainly, we know that variation in antibody genes profoundly impacts vaccine responses and in responses to infection. And there's a lot of good work being done by a postdoc in my lab, but across the continent and many other labs, defining gaps in our knowledge of African genetic diversity. Elaine, who's the postdoc whose PhD was focused on this in my lab, found incredibly high levels of undescribed diversity in antibody genes. About 2/3 of the sequences she identified were unique, had never been reported in any of the databases. And as I mentioned, we know this shapes the way we respond to vaccines. So I think a key gap in the field. It has to be done extensively across the African continent, and it has to be both at the B and T cell level. But we do need to have a much better understanding of what our immune responses to pathogens and vaccines look like. I think the other area where we probably need to put more energy is speaking again about children, but here in the context of vaccination. I mentioned that in adults who are infected with HIV, only about 1 in 10, 1 in 20 of those individuals might eventually make broadly neutralizing antibodies to HIV. But the scenario in children is very different. And this is work from 3 different countries across the African continent with collaborators who are listed down there at the bottom. And you can see that if you look at children aged kind of 4 to 10, the proportion of those children who develop broadly neutralizing antibodies is like sky high compared to what happens in adults. Somewhere between 50 and 80% of those kids will make broadly neutralizing antibodies. It's not quite as great as it looks, because remembering the progression in kids, this will be the half of kids who have survived in the time that at the time, these were half of the kids who had survived in an absence of antiretroviral therapy. And there are many reasons that could explain why higher levels of antibodies are found in kids. Some of them are more amenable than others to harnessing for vaccination. If it is that children have immune systems where the tolerance is not yet matured, and those funky antibodies that I've mentioned a few times are somehow able to better get through and mature towards breadth, then that is something that's very amenable to use in vaccination. But if it's simply the fact that these kids had very, very high viral loads and very good CD4 counts, then that's obviously less amenable to using in vaccination. But there's only one way we're going to know, and I guess that's my plug, is that I do believe there's an opportunity here is to leverage on the years before sexual debut for kids to receive the vaccine potentially really early on, when their immune systems are more amenable to making broadly neutralizing antibodies, and to do so before they become at risk and sexually active. And that's supported now by emerging data from non-human primate studies where neonatal macaques mount much better responses to vaccination than their moms do. So I think another gap for us, and of course this goes way beyond HIV and could potentially apply to all sorts of other pathogens as well. The other place where I think there've been some big strides made in the HIV vaccine field is in the area of slow administration of immunogens. Again, going back to my first slides where I said very few people eventually make broadly neutralizing antibodies. But if you recall, I said that the major thing that drives that is high viral loads. And that's, of course, not surprising for B-cell immunologists, where that constant exposure to antigen over prolonged periods of times it would be much more likely to drive high levels of somatic hypermutation. And so that's the basis of this idea that slow delivery immunization or sequential doses of vaccine may result in better antibody responses in vaccinees. In the non-human primates, use of osmotic pumps to systematically and gradually over a period of days or weeks to administer the same amount of vaccine that you would otherwise give in a bolus, a single vaccine, did result in much better antibody responses in those macaques. And more recently, data from the HVTN looking at the same concept in humans has also shown that there may be significant advantages to priming this way. I'm not suggesting that we attach osmotic pumps to our participants in clinical trials, but where we can make a contribution is, of course, asking people to come in for a vaccine series where they receive the same amount as they would have received in a single bolus over 3 or 4 or 5 very small vaccines in the first week. And that's exactly what people are doing. And again, a gap where I think the significant clinical trial expertise across the African continent could be leveraged to make really meaningful findings here. This is another concept that I feel might accelerate the HIV vaccine field significantly. This is moving beyond germline targeting and testing, what we refer to in the field as the polishing immunogens, the next set of immunogens that we think are required to drive somatic hypermutation. Until now, we've always tested those in HIV-naive individuals, and that slows the field down enormously because it's very difficult to get to the point of knowing whether these vaccines are triggering the right kinds of responses. And as I mentioned, even in germline targeting vaccines, we have to use next generation sequencing where it takes 6 months to get a readout from these individuals. I think that if we were to take individuals who have been living with HIV for many years, are clinically well, are on antiretrovirals, and we were to vaccinate those individuals, then we're likely to get a much quicker readout on whether vaccine candidates look promising, whether they should advance. The idea is that people living with HIV are about as well primed as they could possibly be, and if those individuals fail to respond to a vaccine, then it's very unlikely that an HIV-naive individual will respond. And I think that'll allow us to pass out which vaccines we should move forward with much more quickly than the current rate of testing. And this is something that we've started doing. In fact, our first vaccinee should be today in the Caprisa cohort, where we are vaccinating people who either developed broadly neutralizing antibodies or didn't. And some of this study, a sister study going on in Zurich. But there are several other studies planned or ongoing in people living with HIV that I think will also advance the HIV vaccine field. The AMP trial, which some of you may be familiar with, was a kind of a plan B trial to test in the absence of the HIV vaccine, whether we could prevent HIV if we had broadly neutralizing antibodies, the whole proof of principle here. And this work, which you may be familiar with, was published now some time ago. And this was the use of a single monoclonal antibody that was infused into people who had high risk of HIV infection. It was a kind of a glass half empty, glass half full trial in the sense that overall protection was not observed. But what we did learn is that if we had enough antibody present at a high enough level and people were exposed to a virus that was very sensitive to those antibodies, then in fact they were protected. So the broadly neutralizing antibodies do work if present before infection, but the bar is much higher than we thought. And thinking all the way back to that germline targeting, that means we have to drive those antibodies even further by vaccination, which is a sobering thought. I think the other thing that's changed is our understanding of the viruses that are being transmitted. And this is, again, a plea for other people to do this kind of work. This is a comparison that we did comparing the viruses that caused infection in the AMP trial with viruses that were much older, about 20 years older, and had previously been characterized. And I won't go through the details of the data, but suffice to say that more recent viruses are becoming much harder to neutralize. So again, that raises the bar. Similar data has come out of the IRB protocols and out of the Indian subtype C protocol. So I think, and also out of US studies. And so I think it's becoming clear that this is a true and generalizable principle. It's not a radical change in sensitivity, but it's something we have to factor into our thinking, is that the viruses are becoming more resistant to the antibodies that we are trying to trigger. So another sobering thing to think about in terms of VARs. And then the other thing that I really feel is an important finding that has come out of the AMP trial, and this is very much driven by Carolyn Williamson and her team at the University of Cape Town. For many years, we've had this idea that the transmission bottleneck in HIV is really stringent and almost everybody's infected with a single virus. And that was a very appealing idea from a vaccine perspective, because it meant that antibodies that had been triggered would really only have to block one viral particle, which seemed doable. That was based on the then gold standard of sequencing, which was called single genome amplification, and was a lot of work for not very many sequences. In the AMP trial, that approach was extended to next generation sequencing, and basically the more that Carolyn and Jim Mullins at the University of Washington sequenced, the more variants they found. So the transmission bottleneck we've learned from those studies is much more loose, much less stringent than we had initially thought. We thought it was about 1 in 5 people might be infected with multiple variants. It seems now closer to 1 in 2 variants. 1 in 2 individuals are infected by multiple different variants. So, and that's within the level of sequencing we have. I suspect it's one of those cases where the more we sequence, the more variation we'll find. So again, a much less stringent bottleneck raises the bar yet further. And allied to that is the fact that we don't know, as I've already alluded to, we don't know what's happening across the African continent. Lots of people in this room are online or making contributions here, but this is a schematic from Werner Schmidt and Carolyn Williamson showing that there are massive gaps in our understanding of circulating HIV strains across the continent. And many of the sequences and subtypes that we do know about are sequences and subtypes that are quite old now. We need to have a better understanding of what's out there and how it's changing in terms of sensitivity to antibodies. So again, a plea for people to not give up on this as a research question. It's really still important. And this is my second to last slide, but I wanted to address the focus that the vaccine field has at the moment on the mRNA platform, and this idea that it may be the end of our design woes. It was, of course, enormously successful in SARS-CoV-2 vaccines, and that's on the left-hand side, Moderna. I can never remember which one's which, but as you know, these vaccines were really, really effective. And they're certainly going to be useful in the HIV vaccine space for us to speed up how we test vaccines and to be able to test more iterations and in more populations. But going back to my very first slide, even the mRNA platform doesn't solve the intrinsic biologic challenges that make HIV so difficult, that variation and the huge level of conformational and sequence diversity, as well as a dense glycan shield that hides the virus. Of course, my whole talk has been about antibody responses, because it's all I ever seem to talk about is antibody responses, but very much recognizing that that we need both the antibody responses and the T-cell responses, and this is one study of many that are out there that shows that that the two synergize very effectively. And I know others on this call are doing work in the T-cell space that will synergize with all the work that's happening in the antibody space. So I guess kind of thinking about where we are in terms of viral resistance to broadly neutralizing antibodies, we need more antibody than we realized. And what is unhelpful is that the viruses seem to be evolving towards increased resistance. Allied with that is the fact that the transmission bottleneck is less stringent than we thought. But broadly neutralizing antibody induction strategies such as germline targeting do show immense promise, though they have a long way to go. And there's lots of good science that I think is working towards a much faster model of testing these concepts in small phase 1 clinical trials. And assessing our vaccines that we have, and we have already good ones in other populations, will provide really important insights for vaccine discovery for HIV and other pathogens, which particularly now is urgently needed. So I'm going to stop there and thank our funders, but more importantly, thank the lab. I think a couple of people are online. They are the most wonderful group of people, and many of them, like Kondwani's crew, are students and postdocs, and I'm very grateful for all their hard work. Thanks, Clive.
Great, the room is clapping and people online are clapping.
That's good.
I'm so happy, Penny, that you've seen the light after all these years. The T-cell
I acknowledged them, Ty, because I knew you'd give me a hard time if I didn't.
All right, let's take some questions here, online or in the room. Any questions for Penny? Yes, we have a person here in the room. If you could just say who you are and.
Hi, good day, everyone. My name is Christian. I'm a PhD student here at Stellenbosch. One thing that I have picked out from this talk, it's quite an interesting talk everyone have given, and then the background and then the establishment of Immunopaedia, is that the whole idea of African-based scientists is something I resonate with, but most importantly, the importance of genetic variation, which that have been mentioned quite a couple of times. My question is, with so much progress that have been made in science and the idea of, or the challenges of emerging infectious diseases in the future, how do you address the gap of this genetic variation, being that we've sequenced genes, but do you now sequence the genome and then to see how we can be able to meet future challenges based on that? But also, how can you be able to attract big companies from Europe to come to Africa, not just building future African-based scientists, but empowering them with the platform and avenue to be able to do this research, health-based research, and address health issues in the context of global challenges like emerging infectious diseases?
It's a good question and a difficult question. I think we're at risk now. It feels like we're at a bit of an inflection point where, and others have referred to this already, where we had an emergency response to COVID that significantly strengthened many of our platforms. I'll use the sequencing platform as an example. We, as a continent, went from next generation sequencing being really available and bioinformatics being a major challenge to a situation now where many of the countries on the African continent have next generation sequencing capability and at least basic bioinformatic capacity, and they have the platforms and the expertise. But we're at risk of losing that because we are moving out of the emergency mode. And I think that's going to be the key thing that we will have to do as African scientists is we have to keep our research relevant. I don't think big pharma's going to invest in a big way, and I don't think they will not invest in a big way in things that they are not going to make money out of. And so we have to keep our research and our platforms focused on issues that are of relevance to our public health situation. And we've got to move away from emergency mode, I feel, and go into a much more strategic view of what we're trying to do as a continent, and make sure that we keep the platforms in place by asking the right questions. And I think that's something that we're going to have to focus, is ask the right questions to make sure that we filter all the information we've gathered into public health responsive and vaccines that we need on the African continent, and that way create the kind of market for vaccines, but also for all the upstream work that happens towards vaccines, all the research platforms that are needed. I think it's a hugely difficult space at the moment with funding challenges, but there are also opportunities here. We may have lost some funders, but many have refocused on the African continent. And I think we need to make sure that we capitalize on that as far as we're able to fill some of the gaps that have come up in the last 4 or 5 months.
Somebody online.
We got a hand online. Dr. Monem, what's his name? Nomende.
Nomonde.
Sorry.
Go ahead.
I think you're still muted.
Hello, how's that?
Yes, we can. Yes,
Much better. Okay, good afternoon, everyone. Thanks, Penny, for a wonderful talk, informative as well. Is there any immunological value in low-grade viremia in a person who's on ART, but and the immune system is holding up. There's this thing about U and U equals U now, where we're targeting less than 50, less than 20 in terms of the viral load. But we're finding that some patients have got this low-grade viremia that doesn't necessitate a change to other medication. So immunologically, what do you guys think about that kind of situation, if I may ask? Thanks.
Yes, thanks, Nwanda. It's an interesting question. There may be immunological benefits to having that low-level replication, in the sense that it will continue to drive antibody maturation, but I would totally, absolutely not advocate for it, because I think the risks of it far outweigh the immunological advantages, which would be tiny and unlikely to be clinically useful. I think the risks to patients of having low-grade viremia in terms of ongoing inflammation are much more significant. But what people are thinking of doing, which I think is where your thinking is going, is using very short-term analytic treatment interruptions to assess whether that transient boost in viremia that happens in people who are taken off their drugs or need a drug holiday, whether that has an immunological impact that informs HIV vaccine design. I personally, I think that the use of those kinds of analytic treatment interruptions may be very informative, but I think that's a very different scenario to what you're speaking about, where people are intrinsically not well enough suppressed virologically. That I wouldn't advocate for at all.